Okay. Well, welcome everyone. Thank you for coming on this Easter weekend to this talk. And there are a couple of comments I want to make before I get started right away. Okay. The first one is I want to mention that what you're going to be hearing today is a preliminary study. I've been working on this over the last couple of months. Mainly, I've gotten interested in evolution. I have attended some lectures by Stephen Jay Gould while I was at Cornell, which were just fascinating. And Gould started talking about how evolution basically exploded in the mid 1800s. People loved the ideas of variation, natural selection. They were trying to find case studies to test Darwin's theories. There was the Irish elk, there was the Jurassic Gryphia. And basically, those studies just were sort of ridiculous. In the case of the Irish elk, you had this big moose-like thing with big antlers that people thought just crushed the animal under the weight, which wasn't true. Jurassic Gryphia was supposedly a clam that quilled itself to death, which again didn't work. So in retirement, I've been looking for organisms that allow testing of evolutionary hypotheses, and I think I've found one, Triarthrus. Um, and there have been a number of studies. This thing has been studied for now 180 years. We'll get into that. But that's what I'm so excited about is that these things are very abundant. It's easy to get data. We'll talk more about that in a minute. So right now I'm working on a science manuscript, hope to get it published within a year. And I'm also developing a curriculum for teaching evolution in K-12 schools and possibly college and university. I also want to mention that this has been a huge collaborative project. I've worked with the American Museum of Natural History, the Paleontological Research Institute, State University of New York System, Cortland, and even talked with Curious George at Caltech. So I think this is the most collaboration I've ever done on one project. All right, so with that said, let's get started. All right. So today we're talking about trilobites. These are things that are related to crabs, lobsters, spiders, and so on that lived during the Ordovician time period about 450 million years ago. And specifically, I'm interested in the functional morphology or how these animals basically survive in the oceans. It's an interesting evolution question. It's an environmental question. And I'm going to be giving you the background in a minute. I do have local chat open, so feel free to drop as usual, your comments, your questions in local chat anytime you wish, and I don't have a problem stopping and discussing them. I think that makes more a natural presentation. Oh, by the way, also, if I'm clicking my slides too fast and I'm not giving enough time for them to res, then let me know that as well, and I will slow down happily. All right, so here's a brief timeline for this trilobite. 1832, a medical doctor, where's tagline? Dr. Green describes the first of this trilobite species. They named it Triarthrus beckai, which is interesting because he did not have a good specimen. He didn't have the head, which is usually what paleontologists like to have in describing new genera, let alone species of trilobites. He just had the body and the tail. But nonetheless, he describes this thing, Triarthrus beckai. Fast forward 1838, James Hall in New York defines a new species called T. eatoni. Then years later, 1847, James Hall decides, yeah, Triarthrus really isn't a good name. We're going to assign all of these trilobites to the genus Calamine that lived during the Silurian, which was totally wrong. These things look nothing like calamitid trilobites in terms of their heads, and they lived during the wrong time period. But again, there's a problem. In 1847, the geologic timescale wasn't really established very well. In fact, I don't think the Ordovician was defined. So Hall's doing the best that he can. 1892, W.S. Valen of Rutgers University is working near Rome, New York, and finds these amazing trilobites. these guys not only preserve the exoskeleton, but you get the antenna, you get the legs. And the picture that you're seeing there is of an actual trilobite that I've obtained from this quarry near Rome that's going to be donated to the Virginia Museum of Natural History. It's a spectacular specimen. If you look at the bottom of the head, you'll even see the eye, which was preserved, which is very unusual. So these things have incredible preservations. Um, Valiant decides that he's going to open up the quarry, and a good friend of his, C.E. Beecher from Yale University, goes in and finds lots of these pilotized fossils showing the hard and the soft parts from 1895 to 1904, and starts excavating from the quarry, sending back lots of specimens to Yale University, boxes and boxes and crates of them, and unfortunately passes away in 1904. So he only has 9 years to really work on these things. And as a result, a lot of the fossils that were excavated by Beecher and Valiant were lost, discarded. It's a shame. But the good news is that there are still lots of these trilobites at Beecher's Trilobite in central New York. My mentor at Cornell University, that would be John Sisney, at Yale University in the 1970s, took X-rays of these pyritized fossils, worked out the entire anatomy of these trilobites, from the musculature, the digestive tract, the reproductive system. It's amazing how much we've learned about trilobites because of the pyritization of these fossils. He further argued, based on his understanding of anatomy, that these trilobites um, also have a relationship to modern day crustacean chelicerates like crabs, spiders, and the branching appendages. And Sisney also felt that because of the branching, if you look at any one of those legs, they are what are called bilabious, which means that they're probably not related to modern day insects. All right, we're getting some questions in here. Sumo's asking, does pyritization mean replaced by iron pyrites? Yes. Uh, what happens is, we're going to get to this in a minute. Um, these things lived in very deep water, and there was this rapid mud flow that comes in and covers them and kills them very quickly. And then what happens is the you go to a reducing environment, getting into a little chemistry here, so I got to be careful with mic and attendance. The iron reacts with sulfur and produces iron pyrites from it. And Beecher's trilobite beds were originally leased by Yale University. They decided they was tired of paying the lease fees, so now that they are privately owned, and I'm working with Marcus Martin, who now currently is leasing Beecher's trilobite beds and still taking some really nice specimens. In fact, the picture you see there is another trilobite I got from Marcus that's going down to the Virginia Museum of Natural History next week. All right, and this fossil is so well preserved that is a classic textbook fossil. You know, you go into Moore, and Fisher, which is what I use as in my education, and you see all of the appendages, the soft body appendages labeled, all based on what we've learned from Triarthrus. All right, so let's go over some basic um, morphology and shapes that we're going to be talking about here. I'm mostly going to be referring to the head of this trilobite. And yes, someone's asking, is that why the soft tissues are preserved? Yeah, it all has to do with this pyritization. So in the central part of the head is the glabella, and the glabella would correspond to our forehead, although in our case, the brain is behind the forehead. In the case of trilobites, it's the digestive tract, the mouth, that's under the glabella, and possibly, I think, the reproductive organs as well. Then you have the eyes and the palpebral lobes. We're going to talk about them a lot. These are arthropods, so in order for them to grow, they had to shed their exoskeleton. And that's typically along the facial suture, which it breaks. there are pits, or I should say, depressions in the glabella called furrows, and then the occipital lobe. Max, the palpebral lobe is located on the side of the facial suture. It's where the eyes were attached. It's just, it's not much the palpebral lobe. It's just this thin line that you see. And it's important because I think the eyes rarely get preserved, but that's what's so interesting about the evolutionary history. So we can use the location of the palpebral lobe to determine where the eye was. And Max, that wingy thing, they're just the lobes of the cranidium. All right, basically, trilobites, the way that it works, they've got it's called the free cheeks and the fixed cheeks. So the fixed cheeks are attached to the glabella, and then there's more to the exoskeleton beyond the facial suture called the free cheeks. And that typically breaks off. You normally don't get them. I got a question. Aren't there huge ones eating sharks? I've never seen a trilobite eat a shark. These things more were bottom feeders, or they could go at, from what I understand, they could go after smaller arthropods like copepods. Marcus Smart was telling me. We've actually got preserved Triarthrus with small arthropods or copepods attached to their feeding organisms. So we're learning a lot about these things. All right, so it's 1926. We're now beyond, unfortunately, Beecher's passed away, and Rudolf Rodeman is a teacher in New York State and becomes a very famous paleontologist for New York State. He literally wrote the book on the geology and paleontology of the Ordovician, well, actually the Devonian too, of New York State. And in there, Ruderman says there are two species of Triarthrus. The one on the left in the picture is Beckei, and the one on the right is Eotoni. So he has resurrected the names. As you recall, Paul didn't like those. All right, I don't want to get into too much detail here. And Ruderman thinks that the shape of the glabella can be used to differentiate the two species. That guy has sort of a square shape, and Eotoni is more of a slender, tall shape. And what's beautiful about this 1926 monograph is Ruderman actually gives data for his interpretations. And Bergman, I'm surprised to learn eyes are poorly preserved because trilobites are. Yeah, if you think about Phacops, you're right. Okay, but a lot of trilobites. In fact, the first ones I collected of Triarthrus as a teenager, I thought they were blind. I thought they didn't have eyes, and it turns out that no, there are other specimens. So, like I said, Ruderman actually gives data to support his idea that the shape of the glabella is important in differentiating the two species. And so what I did was, unfortunately, Ruderman only gave data on about 10 individuals for each. So what I did was I doubled the number of specimens. I said, I'm going to look at 20 for each. And it turns out that if you look at the, whoops, go back. If you look at the ratio of the length of the glabella to the width, there's no difference between the two species. It is not a good way to differentiate this species at all. But it turns out they are valid species. They just shouldn't be defined on the basis of the shape of the glabella. We've got to find other ways of doing that. Well, it turns out that we go fast forward to 1983, 1994. It turns out there aren't two species of Triarthrus. There's a total of 6. In fact, there are probably even more than that that have been defined. Some of these things start to develop really weird shapes. Some develop spines that we'll get to in a minute as to maybe what their purpose was. They start changing the shape of the glabella to a rectangular shape. But this pair, Ludvigsen and Tufnell, they work in Canada mostly, and they find that there's a lot more species. And the thing to focus on that these two guys come across is that it's all about the eye. If you cam in to this diagram, you can see that Beckei has a really small eye, And then as you go to the other species, the eye gets longer. And in fact, Ruderman, even in 1926, said there's something going on about the eye and the facial suture. He didn't make a big deal out of it. He focused more on the glabella, but Ruderman was on the right track. The problem was he just was working 100 years too early. All right, so let's talk a little bit about geologic time. Curious George asked me to put this slide in. Trilobites lived from the Cambrian through the Permian, and the Triarthrus ones that I'm interested in were during the late Ordovician time, from about 423 to 488. So that'll give you an idea about where we are in terms of the geologic timescale. All right. So one of the things that's really nice about Triarthrus is that we have really good time controls on these fossils. And as you know or may know, typically the way that we determine geologic time, like we saw on the last slide with the geologic time scale, is using radiometric dating. These are radioactive elements that break down over time. We know that the breakdown of radioactive elements to their stable byproducts are independent of things like pressure and temperature. It's a fixed rate. So they're like clocks that are built um, into rocks. And the number of rocks, mainly igneous and metamorphic ones, that give us a good estimate of the age of the individual rock. The problem is doing radiometric dating is time consuming. It's expensive. I looked into it once. It was like $2,000 a sample, and just didn't have that kind of budget or money. So what we do is we use index fossils. These are fossils that have a very wide geographic extent, but they lived only very short geologic times, and that could be 1,000,000 years. So the idea is you date an index fossil, but every time you find a specimen of that index fossil, you know how old the rocks are. And in my case, we use graptolites. Okay, and here's what a graptolite looks like. They sort of superficially look like jellyfish, although they're not. These things float on the surface of the ocean. They're actually colonial, and we find their fossils very commonly mixed in with the trilobites. And so it makes them ideal index fossils for determining geologic age. Ruderman actually identified 9, what are we called, biostratigraphic zones based on graptolites. Subsequent work has merged a lot of these zones together. We're down to only 3 biostratigraphic zones. And here they are, if you're really into Latin names, these are the 3 main species of graptolites that are used to determine geologic age. A lot of people don't care about the Latin names, so I'm just going to call them zone 1, 2, or 3 as we go through this. But realize the important thing to realize is that the zone 1 graptolite is the oldest and zone 3 is the youngest. So keep that in mind as we're going through this talk. All right, here's a little here's a summary of the geology that's going on. Tagline is saying, did the upper dome have an air cavity? I don't think so. These are marine organisms. So they had gills. They did not have lungs. Um, they they're usually compressed and flattened in the shale. Although we do have some that do show the 3D geometry. I think that was just for the soft part anatomy. But it turns out, again, Taylor, you're a little bit ahead of me. It turns out that the gills are not inside the exoskeleton. Can be extended outside to the limbs, and that's important. All right. So anyway, like I was saying, this shows, for those of you interested in geology, and I don't know if you are, the main lithologic units. So we got the Dulles-Rill, Indian Castle, Utica, the Whetstone Gulf, and so on. Geologic formations are given names just so to keep track of them. And I've put on here the Canadian units as well as the New York ones. All right, so let's talk about plate tectonics because that's going to be important. 450 million years ago, there was a volcanic island arc off the coast of North America called Laurentia. And as this volcanic island arc is shoved against North America, you produce a deep basin along the margin of North America. And then sediment is shed from North America and also from this island arc, filling the basin in. In fact, a good way of thinking about this geography is not North America today, but think of Japan off the coast of Asia. That's what North America was like 450 million years ago. And that's the basin that these trilobites lived in. It was probably very deep. The oxygen levels were very low. Um, you'd think it would not be a good place to live, but it turns out it was for these trilobites. All right, like I said, the older tribes, the Beckeye, probably was able to hide in the deep water regions. In fact, that's one of the advantages of a deep basin. And probably that's why they didn't really need good eyes from predators. And there were quite a few in this basin, a lot of squid-like organisms that we're going to get to. But as the basin gets shallower, now we've got a problem, and that is the photic zone will start extending into the basin so the predators can find prey, namely Triarthrus, more easily. So that's part of the story that we're going to be doing today. All right. So the nice thing about living in deep water is that it's easy to hide from predators. Also, what happens is plankton and other organic matter settles to the bottom. Triarthrus likely was a deposit feeder or fed on small arthropods called copepods. It's an organic rich environment. But it is also a low oxygen environment. And the latest work by the American Museum of Natural History indicates that there were gills attached to the appendages. So what Triarthrus did was to extend the appendages and the gills in a fan shape on the bottom of the seafloor to try and maximize the respiration that's in these environments. Normally, when you get in very deep water environments, the only things that are able to extract oxygen are things like worms. Seem to do a good job on it. But it seems like 450 million years ago, Triarthrus figured out how to survive. And they did well. They survived for 7 million years in this basin. Okay, this is a phylogeny or family history. Again, you may need to cam in to see this diagram. Sorry, the print is so small, but the ancestor is T. beckai, which gave rise to T. eatoni. And then from T. eatoni, you have a series of other species like Spinus glabella, Canadaensis, and so on that arise from eatoni and beckai. Right. So, like I said, I was able to get data from the American Museum of Natural History, the Paleontological Research Institute, and Dr. Chris McRoberts of the State University at New York. Most of the data that I've obtained has been from the central part of the U.S. The gray pattern that you see on the map is the outcrop for the late Ordovician, and then each dot represents the main localities that were sampled, mostly by SUNY at Cortland, Chris McRoberts and his students. And shout out to Chris, I know he's not here, but he's been a tremendous help in this project in sharing his data and suggesting places to go to look for these things. About a couple of weeks ago, I had about 100 images of Triarthrus that I had analyzed, taken measurements, and also ran through the computer. Uh, Last week, I was in upstate New York and met with Chris McRoberts, and he allowed me to go through his collections, take pictures. So I'm now up to about 200 images. I just haven't had the time to go through and analyze it. Although I'm not sure it's going to matter much because it seems like the patterns that I'm seeing are very robust. As it went from 50 to 100, really didn't see much of a change in my conclusions. But to be more thorough, I want to get more data as we go along. Also, notice on this chart, uh, beckai and eatoni are the two most common species. And then you get a spattering of some of the other species. So the data is strongly biased to those two species. We know a little bit about the others, but not as much as we'd like. All right, so now I'm sure you all know that evolution proceeds through variation, natural selection. And one of the techniques that I've been studying for the last couple of months is called landmark analysis to measure and detect changes in variation and natural selection. Geologists, because we have that deep time, we can take a look at time spans and how organisms are changing over time. And you may be familiar in 1972, Niles Eldredge and Steve Gould challenged typical Darwinian thinking. Darwin said that there's this slow, gradual change of one organism to another. Eldredge and Gould said, well, not so fast. We think that organisms or species stays the same over time, and then there's this rapid shift in the shape of the organs or the morphology. And the beauty of Triarthrus is we have such good time control and geographic control that you can actually take a look at which mode of evolution is occurring. We can test for stasis and so on. And I'll be saying more about that in this talk. All right, so here's the way that landmark analysis works. You take a picture of your trilobite, you superimpose an XY axis on it, and then what you do is you click on certain points on the trilobite, and the computer will record the XY coordinates for every time you click. This would be a very slow, tedious process if we didn't do it with computers, but it goes relatively quickly, and you can go through about 100 species in these landmarks. And this is what the data looks like. It's just a text file, so it records the X and Y coordinates. You'll also notice at the bottom there's a scale value. So you have to set the scale so you're telling the computer basically how many centimeters are in a pixel, I think is the way that it works. So that'll be important because one of the things that landmark analysis assumes is that it really don't really don't care about the size of the specimens. That's so distracting. You want to notice if there's any change in the shape. So landmark analysis will normalize all the specimens to a unit size so you can just look at changes in the shape. And so here's the result of a landmark analysis. This is for 22 T. Beckei fossils that I ran through the program. And you notice that there's typically a circular scatter around the mean here. Sumo's asking, oh, what time scale? What is fast and what is slow? In my case, I'm looking at a 7 million year time scale, Sumo. Typically, species change on the order of maybe a million years, something like that. Maybe a couple of million years would be slow. And Vic's trying to answer that as well. Very good. Okay. All right. So on this slide, what I want you to notice is the third landmark. Okay. This corresponds to the rear edge of the eye. And the pattern of landmarks for those 22 specimens is not like the others where it's like a circular shotgun blast around the mean. Okay. Instead, it's skewed out. It's almost like a teardrop shape. And I think what we're seeing here is even within one species, there's this migration within that population to have the eye get larger and also move towards the posterior or the rear end of the cephalon. So I think we may actually be seeing natural selection as well as variation within this species. And here's another way of doing it. I can eliminate all of the dots for the landmarks and just show the averages. And also I put on vectors here. So the two main landmarks that are moving are 2 and 3, which correspond to the front and rear part of the eye. It seems to be what's going on here. There's a little change in some of the other things, but nowhere near as marked as the features of the eye. And Melanie Hopkins at the American Museum of Natural History encouraged me to play around with deformation grids. I read up on these. I'm not happy with them. I think they're a little confusing. But basically, the way that these work is look at the grid. If the grid lines are nice and horizontal or vertical, then not much change is going on. If you look at landmark number 3 that's labeled on there, that's where you see the most deformation in the grid. So that's where most of the evolutionary change is occurring. I'm not happy with this graph, although it is commonly used in paleontological manuscripts. Like I said, Melanie encouraged me to put it in there. I actually liked the previous slide with the vectors. I thought that was more meaningful to me, but uh, whatever. All right, so the next thing that I did was I took the landmark information and fed it into a principal components analysis. The idea here was to reduce the dimensionality of my data. I had about 8 landmarks in my measurements. And so what principal components analysis did was to reduce the variation to just 2 axes, PCA 1 and 2. And that's what we see here. And don't even try to read the little numbers there. I made the print way too small, but basically what I saw was that um, the the first axis, what it did was to pull apart the species. So you have Triarthrus becki, the ancestor, on the right-hand side of the first principal component axis, and it put Rogensis, which is the youngest species, on the left-hand side. And then the second axis pulled apart some of the species that are in between, things like, I believe it was Canadaensis, and one of the other two separated from Eotoni. So it was pulling apart the middle age species that are there. And again, I think I didn't actually tell the PCA, I didn't give it the age or the species. It's basically working off the morphology, changes in the eye to differentiate these specimens. For those of you that like PCA analysis, this shows the variation accounted for. The first axis accounts for about 37% of the variation. The second is about 23. So the principal components analysis is picking up somewhere around over 50, 60% of the variation within these measurements. And then once you get past the second principal component analysis, the amount of variation drops off quickly. So further axes that you could look at, we're probably just looking at noise or changes that aren't very significant. All right, so basically, what does it all mean? Uh, what it all means is that it's all about the eye in these guys when it comes to evolutionary changes. And the cool thing about trilobites is they're living almost 500 million years ago, and they're developing some of the most sophisticated optical systems. I was just really impressed when I started to read over the literature on trilobite eyes. You have very so-called primitive organisms living on the bottom of the ocean, and um, what they had to do to create their eyes was to extract calcium and carbonate ions from the ocean water and create lenses of crystals of calcite. And they then arrange them into a dense packing of hexagonal patterns on the eye surface. And they don't do it randomly. They arrange these calcite crystals so that the long axis of the crystal is perpendicular to the optical surface. This is brilliant. If you know anything about calcite, if you look through a calcite crystal at the wrong angle, you get a double image. It's hard to even focus. But if you look along the main axis, what happens is that the two images merge to one. And if you worry about capturing prey, like a copepod or evading a predator, that's crucial. So what I decided to measure was the size of the eyes, the field of view, and I did that by drawing in lines perpendicular to the surface of the eye and seeing where they intersect. And I also measured what I call the azimuthal angle, which is just a measure of how much the eye is rotated from the middle part of the glabella. All right, so what I basically found is, first of all, the field of view increased from 56 to a whopping 180 degrees. Humans are only 90 degrees, which is just crazy that these animals were able to develop eye systems that had twice. Yeah, birefringence is the fancy term tagline. Calcite has very high birefringence. Um, so definitely they're increasing the convexity of the eye through geologic time, trying to maximize. It's like the lenses they put in virtual reality goggles. So you're trying to magnify your field of view. So yeah, the lenses, they're sort of like eyeglasses, except our eyeglasses are made out of plastic or glass. Those were entirely made of calcite. And don't think of them as one lens like we would have on our glasses. Think more like a B. It's a compound eye system of a whole bunch of smaller lenses. One of the things that I found is that the size of the eyes are directly correlated to the size of the head. So we're absolutely seeing some growth stages as we go through these trilobites. And that's important to take into account because if you just try and plot eye size versus the species, you don't get meaningful information. But if you compensate, if you divide the length of the eye by the length of the cephalon, you see a very clear pattern where T-Beci, the ancestor, had the smallest eye to cephalon length, 0.23, and then there was an increase to T. toni and rogensis. The eyes definitely were getting larger as you go through geologic time. And Vic, thank you for throwing up a website. So there's an increase in the field of view. There's an increase in the eyes. And I also wanted to look and see if the eyes were rotating around at all. So I measured the angle of the eye relative to the center of the glabella. And sure enough, there was a change. And I decided to use a polar coordinate graph to try and show that. So T. Beckai had an average rotation of the eyes about 65 degrees. The toni was about 70 and rogensis was about 75. Good question, Max. Are you asking, are there more lenses in the eye as they got larger? I don't know. I presume there would be. I would think the lens would be about the same size. The problem is that the lenses are very small. And yeah, it is definitely a compound eye. There's a preservation problem, and the other issue, there's a visual problem. I've got a really good eye, and I want to take it down to the museum, put it under a good high-powered microscope, and see if I can see the lenses. I think I probably can, but my feeling is that I'm probably not going to see many of those lenses because in the specimens I had, it's rare that you even get a good eye preserved, let alone the lenses to it. All right, so the point is that as you go through time, this animal is rotating its eyes by about 10 degrees. Um, and of course, there are animals today like chameleons that can even rotate their eyes even more than that. But I think what's going on here is that it's doing everything it can to improve its field of view. By the way, one thing that you'll notice on there, those bars show the standard deviation around the mean and also the minimum and maximum. And in the case of T. beckai, there were some specimens that I measured that had a rotation of 78 degrees. So within the T. beckai population, there were individuals that could rotate or create an eye rotate more or equal to rogensis. It was just the mean of the average chose not to. I'm not sure why a small rotation would be advantageous evolutionary. I'm just saying that that's what I'm seeing here. So it's variation natural selection. I'm assuming that basically the individuals that had more rotation were selected out and the others were eliminated. All right, so what's going on here? I think what's going on basically, and Rudiman even got it in this diagram, is if you look at the body of this trilobite, the pleura, uh, thorax on the side is complete, but on the left side, it looks like there's missing parts, and it almost looks like chew marks that are there. And there were over 40 different species of cephalopod swimming around the waters. And if you look at modern day squids, they are extremely aggressive predators. They're smart, they're fast, and I think the same thing was going on back in the late Ordovician. These things were running around the bottom of the seafloor looking for arthropods, but back in that day, it was trilobites they were feeding on rather than crabs and shrimp that you see today. So, what's driving the evolutionary changes, I think, are predation predators. So, anything this thing could do, make the eyes larger, increase the field of view, rotate the eyes towards the side, would give them an evolutionary advantage to other trilobites that didn't have those abilities. And here's an example of what the squid-like predators would look like. Back in the late Ordovician, cephalopods had shells, so they probably weren't as agile as modern-day squids are today, but they certainly were carnivorous. All right, so one of the things that I've got to worry about is how do you tell the difference between taphonomy or normal breakage of shells versus predation? That was a question that was brought up when I was at the American Museum about a month ago. And I said, I think the answer is obvious. We do know the trilobites were arthropods. They had to molt their skin in order to grow or through their life cycle. But again, if you cam in here, what you'll notice is that when the exoskeleton breaks apart for molting, the tail will break off from the thorax. That's a plane of weakness. Thorax breaks apart from the cephalon, and within the cephalon, the individual segments will split apart. That's where the plane of weaknesses are in these trilobites. When you look at predation, squids basically cut across the hard parts of the pleura and forms like a semicircular mark within these trilobites. So I think it's fairly easy to recognize the difference between predation versus normal breakage within the trilobites. And there have been a number of studies, not so much of Ordovician predation, but certainly um, predation on Cambrian trilobites is very well documented in the scientific literature. So I basically worked off of that to try and recognize predation in Ordovician trilobites. And so here's some other examples from the published literature, which looks like there might be bite marks. The guy on the right has pretty much lost half of its tail, even though it had spines. So the spines may have been a deterrent, but it wasn't totally effective. And here's some more bite marks. By the way, you notice that there are spines coming off the head. And I was talking to Marcus Martin about that. He said, I doubt those spines were a deterrent for predation. He thinks that by extending the spines out away from the head, they might have gotten more stability on a soft substrate, which is a possibility. Maybe it's both of them. And here's a weird fossil that I found. You would think the spines would have been hard and rigid, but evidently some of them were not. Maybe they were able, I'm not sure if there was a muscle attached to it, but it looks like they were, in some cases, flexible and were able to move from side to side. Again, not sure what the benefit of that would be, maybe to just visually deter a predator. All right, so here's a little summary of what I've said. We see a rotation of the eye. Now, when it comes to phyletic gradualism, punctuated equilibrium, what I've noticed is that the amount of rotation of the eyes for Eotoni stayed about the same, about 70 degrees. So I think what happens is we're looking at a clear case of punctuated equilibrium in these trilobites. So when a species gets established, it pretty much maintains that morphology through its life cycle, and then it rapidly gives rise to a new species, and then they stay stable as well. All right. Tag, I'm not sure what you're saying. It might have been operative conditioning in the primitive nervous system. Again, I'm not sure if the spines would have been controlled by a nervous system. All right, so here's my conclusion. Eye size increased over geologic time in the Ordovician. Um, species learned spines were not pleasant. I'm sure about that. Field of view increased. Eyes were rotated to the eye of the rear of the head. And this may have avoided predation in the past. All right. So, like I said, I'm working on a manuscript, trying to put all this together and get it published this year. I'm also working on a curriculum for teaching evolution in the high school classroom. And you notice I put a red box beneath my slide projector. Like all of you to click on that, you'll see a link to my slides. So if I went this a little too quickly, then I see some people clicking already. That's great. Feel free to flip through them, through the slides again. Also, you'll see it's got my email in there. It's also got a download link if you want to take a look at this curriculum that I'm working on for K-12. You can download the entire curriculum, and it's got about 9 lessons in there already. And the idea was to try and simplify a lot of the techniques that are used in modern day paleontology to a point where students could understand it. For example, landmark analysis can get a little tricky and complicated, but you can actually put these out and you get dot to dot puzzles, which show clearly evolutionary changes that elementary school kids can pick up on. All right, so we got 15 minutes left, and over the last week, I dealt with the question of how do I get more data? 100 specimens really isn't that much for doing morphometric analysis in paleontology. Most studies have hundreds or even thousands of specimens that are analyzed. So last week, I took a field trip around New York. I went to the Paleontological Research Institute near Ithaca, New York, and was given access to my advisor's collections. So there's John Sisney made collections of thousands of fossils when he worked at Cornell. Each one of those dots represents a sample on his correlation diagram. And I went to the PRI, and they actually gave me access to his field notes along with his other writing and figures, which I was grateful about. Ultimately, those black field notes need to be scanned in. I hope to make a PDF out of them. Um, I also was allowed to get into the storage area courtesy of Bolt Storage in Ithaca, New York. These specimens that Sisney collected are in a climate controlled environment. I was happy to see that. So they unlocked the doors, and here they are. There's hundreds of small boxes of samples of shale with fossils in them from John Sisney's collections. Most of them were sealed up with tape. And unfortunately, that day, I didn't have a lot of time to go through all those boxes. One of them, sample 260, has recorded 1,600 specimens of Triarthrus in it. So I'd really like to find that one. My concern is that a lot of this material might be fragmentary, perhaps too, operative. I was impressed when I saw 1,600 specimens from one sample. All right, and here's what it looks like when you take the box apart. You see uh, just samples of shale. I looked through it and really didn't see many fossils there. And I talked to the folks at the PRI and I said, I hope that you're able to move this collection back onto your site so it's more accessible. And they said, well, it's all about a amount of money. They need about $1 million to expand their area, and they've been struggling. I made a donation while I was there. But I'm hoping that it's a great facility. I hope that they are able to raise more funds to move this stuff back in. After that, I visited Chris McRoberts at Cortland, and for Mike Shaw, I put a flume hood in just to introduce a little chemistry to wake him up. And the reason why they had the flume hood there was they had a Bunsen burner and a container, see a little air pump there, where you could put ammonium chloride in there, and I bet you like that, Mike. Um, and you could heat up the ammonium chromite, and then there's a air bulb that you blow it onto the specimens. So you can turn them from black to white to help pull up some of the specimens on the fossils. This is the first time I got a chance to try it, and it was pretty cool. And then on the opposite side, away from the flim hood, was some really nice binocular microscopes, a full photo lab set up, uh, and it was a great setup for taking pictures. And Chris grabbed a lot of his drawers of Triarthrus for me to go through and photograph them. The other thing that Dr. McRoberts gave me was this correlation diagram showing about 9 or 10 of some of the best places in New York to go collecting these fossils. And so for the rest of the week I drove around, it was the tail end of winter. So it was really hard to get access to some of these. They're mostly creeks. So instead, I just tried to get permission because the museum doesn't like me going onto private property without owner permission to collect stuff. And here's some of the places I want to go over the next couple of months. This Canajoharie Creek, it's stop number one. It's some of the oldest Triarthrus fossils that have been found in New York. The problem is that it's on the bottom of steep gorges. So you walk along the path and you look and, oh my gosh, it's like a 300-foot drop. And it's the end of winter, so all the snow and ice is melting. And where does all the meltwater go? It goes right into the creeks. So the current is extremely quick. And exactly, Mike, get permission is my mantra. And unfortunately, Canajoharie Creek is owned by the town of Canajoharie. So I've got to, next time I go up there, which will be May and August, I'm going to stop by and see if I can get permission to just drive along the surface line. Although the rivers pretty much are public. All right. But if you want to see what a fossil looks like from Canajoharie Creek, here it is. It's a typical T. Beckii, and as you would imagine, a very small eye attached to it, but it's still there. So I was able to get some specimens from Chris about these very old Triarthrus. And the other thing that I'm doing is trying to educate the landowners. Here's Noandaga Creek. This is in the middle of the sequence. And so I just started knocking on doors. And it turns out that one of the landowners for Noandaga Creek is an amateur paleontologist. Ever since she was a kid, she would walk along the creek and find some beautiful fossils. And she let me take some pictures. And she said, feel free if you want to walk along the creek, go for it. So I did. I didn't find any fossils, but always nice to get permission from. And this benefits both of us, right? Because I'm getting access to the property that has the fossils, and she's being educated on what's there. Like I told her those were 450 million years old. She didn't believe me. But anyway, Mr. Earl, kudos to you. Um, Chris also suggests I go to Asquago Creek, and he goes, ah, there's barbed wire. Just be careful, crawl over the barbed wire. And I'm going like, no. So it turns out it's farmland. And I tracked down the owner of Oswego Creek Banks, and they said, no, I can't give you permission to go on that area. I'm afraid somebody's going to get hurt. There are fast currents, you slip, you're going to get into serious problems. But there's a grange near there that did give me access. Do you have any experience in Middle Texas area? No, I don't, Andre. I have been to West Texas. I haven't been to Middle Texas, and my suspicion is that knowing the geology of Texas, you're probably not going to find any trilobites or Triarthrus because it's mostly Cretaceous in age, especially around the Austin area. The rocks are just too young. Threaten their life with a trilobite, ready to shoot you. Okay. Yeah, my nephew is a New York City police officer, and I asked him about trespassing, and he said, no, if they call the cops is what they should do. The cops will make you leave the property. I don't think New York is a stand your ground state. I know Florida is. I don't really care. I'm really careful when it comes to trespassing. I get permission first before I go anywhere. All right, so here's some good news. One of my favorite spots I want to go to is Daldrill Dam. Look at all that shale and the pretty water flowing over it. Um, and I called up the manager for Dulgeville Dam and got permission. They said, sure. In fact, you could have gone on the property when you were down there. And I said, nope, I'm going to ask permission first. So when I go back in May and August, Dulgeville Dam is the place I want to go. I mentioned to you, John Slisney had a sample with 1,600 trilobites. It came from Dulgeville Dam. So that's high on my list to hit that site next month. Another place I want to go to is Sugar River in Constable, New York. There's a public baseball field, and you can see it on the left-hand side of the diagram. So the plan is to walk over there. That should be public property, and walk down to the creek. And there you see one of the fossils that Chris collected. Beautiful specimen of a Eutone with the glabella, the free cheeks, the fixed cheeks, the eye, at about half of the thorax. Just a beautiful specimen that you see there. Better than it is. It really is a matter of, I think, just a matter of, you know, plate tectonics and the fact that, we have basins forming in the Paleozoic that allowed seawater to come in and preserve it. All right, if you look at the top of the diagram, there's the Whetstone Gulf Formation, and I would love to get Triarthrus. They are in that formation, and the problem is access. The Whetstone Gulf is part of Whetstone Gulf State Park, so I applied for a permit twice from New York State, and twice I was denied permission to go into Whetstone Gulf and collect fossils. So the only thing I could really do as a tourist is to go in there and look around, but they wouldn't even allow me to publish pictures of any fossils, which I think is ridiculous. What I'm planning on doing is visiting a paleontologist in the state capital, Albany, and see if I can work through her to get access to Whetstone Gulf, because I would really like to see what's going on. Or go up to Canada. I hear that there are very young Triarthrus up there. And here, because Mike likes to throw up pictures of cats, I decided to end my talk with a picture of my cat. And of course, when I went to the PRI, I got her a Trilobite toy to play with, and I put some catnip on it, and she really does love, I should say, he loves his Trilobite toy. So, yeah, Wilson. Um, so anyway, uh, stay tuned. I hope to come up with an update on this. I'm just having fun collecting more data on Triarthrus, and I hope to come back later on and give you an update. But to be honest, a lot of the patterns that I'm seeing in Triarthrus are very robust. I think doubling, tripling the amount of data. I don't think it's really going to change my conclusions, but like I said, I will come back and talk when I have more. Absolutely, Max, be happy to. I've got a manuscript. I sent it through Melanie Hopkins at the American Museum, who said I would accept it. She wanted a couple of changes, which I have made. So I think getting it through peer review is going to be easy if I've got someone like Melanie watching my back. I talked with Curious. Many of you know Curious George. He wanted some changes. He wanted more data, but he said he would work with me. In fact, I'm really looking forward to what a Caltech professor would recommend in terms of data analysis. I think this could be a real boom for paleontology. And that's it. It's 2 o'clock. Bam. So I can stick around for a few more minutes. Anybody else got any questions or comments? Although I think that that this pretty well does it.